So Dr. Larry Mellichamp is a retired botanist from the University of North Carolina in Charlotte. He's an award-winning professor and author. He was also the director of the Botanical Garden at the university. He is renowned for his expertise with Sarracenia, and the title of his talk is How Do Sarracenia Avoid Eating Their Pollinators? And I'd also like to say he brought some Sarracenia that are in the sale area over there.
Thank you. Put this back on and see if it's working okay. Seems to be. And I have to get the... So my talk today is not going to be technical. We can turn the lights off, and we can watch the slides. I'm from North Carolina. You may have noticed all the bullet holes on the signs. And this, by the way, if you've never been there, is kudzu. If you ever go east of the Mississippi, you will be engulfed with kudzu. But it doesn't grow in pitcher plant bogs. So I'm going to talk about very low-key things today, about how just some musings on how pitcher plants avoid their pollinators, or hey, don't shoot the messenger. But first I want to say how proud I am to be at this conference and to see all the people here, people of all ages, all stages of their education from all over the world. When I first started being in Carnivorous Plants 50 years ago, you know, all three of us would get together and talk as best we could. So you all have been at the beginning of what will continue, I think, as a golden age of carnivorous plant science that did not exist before about 1976, with probably the beginning of the Carnivorous Plant newsletter was the beginning of the coagulation of people and ideas. I was in Don Schnell's office in 1974 when he said, we're going to start a Carnivorous Plant newsletter where 10 of us are going to correspond across the country. And because we can't seem to communicate very well, so we need a better way to communicate. And of course, that exploded into hundreds of people from around the world, and who knew? So folks have asked me how I got started with carnivorous plants, and it's as many of you did, although back in my day there were no books, no internet, no mentors, no botanical gardens, there was nothing. And I killed my first Venus flytrap, as everybody has, when I was about 10 or 12, by putting it in a window and feeding it hamburger. And so I have told you've all told countless young people, don't feed it hamburger. Give them sunlight. But I really got started only as a college student, believe it or not. So many of you got started right out of the cradle with carnivorous plants. But I was a college student, and my professor took me down to the green swamp, famous green swamp of North Carolina, where we have so many different kinds of carnivorous plants. And he said, go find a Venus flytrap. And I thought, well, that'd be easy. So I spent all day driving up and down Highway 211 trying to find a Venus flytrap. I knew what they looked like, but I didn't know where they grew. And so I tramped over hill and dale and never did find one. And as the day ended, I was quite sad. And just as I was getting back in the car with the final rays of sunlight, in fact, I think it was the headlights of the car, actually, down in the ditch by the car was a large Sarracenia purpurea. And I recognized that, and I said, here's something. So I actually dug it up out of the ditch, took it back, and that was my first encounter with carnivorous plants. And then the rest, I didn't know I was going to become interested in them, but things happen. And as all of us, we're products of the people that we encountered and the mentors and the opportunities to deal with carnivorous plants. And so I happened to come along at a time when I had people who influenced me and showed me how to take this road to study carnivorous plants, and especially Sarracenias, because I live over there where they all grow. So I'm going to start out today with just a few things from from back east, and I want to show this first slide. So I took this picture 20 years ago, and I believe that these are the local signs of the Bay Area Carnivorous Plant Gang. This must be the way you mark your territory. I don't remember where I took this. I believe I was in probably downtown San Francisco somewhere. So I'm sure that was a sign of the future to come. Now, back east, something has happened remarkably in the world of art and carnivorous plants, and this may be a first. I'd be interested if any of you all have seen anything like this. In Charlotte, North Carolina, we have just gotten a 12-mile extension of our light rail. It goes from uptown out to the university. And this is all built over the past 5-6 years. And as they were building it, I noticed something unusual about it. I noticed the concrete facades along the edge of the rail line, and I noticed the abutments holding up the trestle. And if you're not busily in traffic and you stop long enough, what do you see there? That is a Sarracenia. The abutment is the hood of a Sarracenia and the tube of a Sarracenia. On the facade of the overpasses, there's a close-up so you can see even more the veins of the Sarracenia. There's the pitcher, there's the lip. I don't know why that little notch is there. That's not very accurate. But this is an artist's rendition. And if you look on the facade, you see strange botanical hieroglyphics, if you will. And that's when they first started doing this 5 years ago, I thought, whose a cocaine-induced or opium-induced nightmare has created these images. And when you look at them up close, there's a sundew, there's a dew pine, there's a utricularia. Who ever saw carnivorous plants in public art? So I think this is a first. And the way it happened is the designer who did this is a person who does, when she does these kinds of constructions around the country, looks for ways of expressing local features. And what happened, I talked to her, what happened is she came out to the university like 10 years ago when she was planning all this, came into our botanical gardens and said, I want to see some books on carnivorous plants. And so it was probably me, I don't remember her, because people come in all the time. So I probably gave her like Barry Rice's, what would then been a recent publication book on carnivorous plants, because these look just like his pictures in there. So you tell Barry Rice that I think his pictures are immortalized in concrete. And that's how she got these ideas. It was by looking through a book, and she knew that North Carolina was famous for carnivorous plants, and that's how it came about. So I think this is the first of carnivorous plants in public art. And then just this past summer, I was looking, you know how fate plays a hand, and you don't know how it happened. I was looking through one of 100 books on the side of a wall, the bookcase, in a friend's old home. And here's a book from 1949, A Traveler's Guide to Roadside Wildflowers, Shrubs and Trees of the United States. And here's a picture of Sarracenia flava in color, 1949, in a book of roadside wildflowers. And you notice there's 700 wildflowers in this book and only 4 color pictures. So here's Sarracenia flava rated a color picture when color was prohibitively expensive, published in a book of roadside wildflowers. And I guarantee you are not going to see any Sarracenia flava that you can see from a car anywhere now in the Carolinas this day and age, but maybe back in 1949. So places like this where you can see a meadow of Sarracenias are not on roadsides anymore. they're all destroyed. So that was pretty remarkable that Sarracenia, I think that's the first publication of a color picture of Sarracenia in a book. Not that wasn't a drawing or painting. Plenty of those in old books, but the first color photograph of a Sarracenia, 1949. So I was pleased to find. When you think about it, you realize that no insects are going to make it across that meadow. Sooner or later, they're going to be stopped at one of those pictures. This is one of the finest hillside bogs over in the Carolinas. It's down in northeastern South Carolina, and it's one of the great remaining places where you can see 5 acres of Sarracenia flava. They burn it every year, and it's remarkable to walk through there. And there's 3 species of Sarracenias. There are no flytraps there, but there are other bog plants. So we're fortunate to have a few places left. And so, you know that Sarracenias grow in pine savannas and flatwoods, and you know that they attract insects. It's not hard at all to find a fly sitting there. You know that these habitats are wet, acidic, sunny, low nutrient, and what's the fifth factor? You have a lot of it out here in California. It's fire. Fire. That's the 5th factor of habitats for carnivorous plants. And I don't know whether you can tell from this picture, that is a miserable place to be for humans. Hot, 95 degrees of heat, 95% humidity, mosquitoes the size of small jets, horseflies the size of bulldogs. If you look around, briars the size of broom handles. If you look around, you can find black widow spiders and water moccasins. I've never seen those. They stay hidden. In all my years of going down there since 1968, no bears, no snakes, no spiders. I've never seen anything dangerous except exactly. So my rule is the more miserable it is for humans, the better it is for carnivorous plants. And all you really need there is a little bottle of 100% DEET to cover your skin. Otherwise, what people see you out in the bog, what they see is your profile as a swarm of mosquitoes moving through the bog. So I want to review a little bit the taxa of Sarracenia, just to remind us of what I'm talking about. And I'm not trying to be controversial or professorial, or I'm not going to impose any opinions on you. I'm just presenting, I guess it is my opinion, but I'm not hitting you over the head with a 2x4. I recognize 11 species and some subspecies, but in the genus Sarracenia, you've got 4 what I call individual species. They don't have any close relatives: Aleda, Leucophila, Minor, and Psittacina. They're species, entities unto themselves. A certain amount of variation in them, of course, but there's no subdivisions. And then there are the complexes, what I call the complexes. The flava complex has Sarracenia flava and Oreophila, that really, they should be, they could be subspecies if you wanted to. They're similar enough. And you've got the Purpurea complex. Purpurea, the subspecies venosa in the Carolinas, the poorly described and yet not well-known form Montana up in the western Carolina, upper Georgia, and South Carolina mountains. Not well-known, maybe extinct, almost. Rosea, the Purpurea down on the Gulf Coast, sometimes referred to as Burkii. So I'm using the names that are familiar to you at whatever level you want to recognize them. And of course, the super controversial rubra complex, with rubra, gulfensis, jonesii, alabamensis, and wherrii. And so I'm presenting these as names of entities without, I'm not going to go into whether they're species or subspecies or varieties or whatever. That'll be for you to, and I'm perfectly accepting of some of the other interpretations, but I just wanted to get the entity. These are the taxa that we deal with at whatever level you want to recognize them. And all of them, of course, except purpurea, are found in the southeast, south of Virginia and through the Gulf Coast as far west as Texas. So who knows what species goes all the way to Texas? Elata. Sarracenia elata really is almost at its best in East Texas, but it's pretty common along the Gulf of Mississippi, Alabama, and Florida, but it goes all the way to Texas. And of course, the one that goes the farthest north is purpurea. And each of these has a range, a defined range, and you can find some bogs. It's easy to find bogs with 3 species in them. It's harder to find bogs with 4. I don't know if you could find one with 5. And whenever you find 2 or more species, they hybridize one way or another. And so sometimes it's hard to find a pure species. And so I just note that the folks studying Sarracenias in the lab at molecular levels and chemical levels, I sort of sit there and wonder, how do they know that the material they were studying was pure, was really straightforward, pure, unhybridized, whatever they said it was? Because it's really difficult even for experts to recognize the species separately when they all grow together. They're all a little bit hybridized. But many of these are isolated. Jonesii only grows up in the mountains, and Alabama Menses is only in central Alabama around Montgomery. Rosie is only along the Gulf Coast. Montana's only up in the southern Appalachians. So each of these, the good news is that each of the controversial ones has a range, and you can almost identify them based on where they came from. If you knew where they came from, you'd be able to know what they supposedly were. Let's see. So I took this beautiful collage off the internet just to remind you of all the different shapes and sizes and variations in pitcher plants. They all have hoods to one degree or another and veins. Some look like flowers, some don't. They may all think that they look like flowers. They certainly secrete nectar around the mouth of the pitchers to attract their prey. And if you have not done so yet, you should find some fresh pitchers, maybe over here in the sales area if touching is allowed, and get some of that nectar, assuming the plants haven't been sprayed with DDT or anything. Take some of that nectar and touch it to your lips and tongue, and you may feel, if you're a sensitive person, you may feel the numbness that comes from something in that nectar, which we believe stupefies the insects and helps them to more quickly fall in, with or without wettable rims. That probably helps too. And here's another little thing. You see how I'm bringing a little sociology into this talk. When I was in Australia at this conference in '08, which was a great experience, but I met an older lady, middle-aged lady there in town. I was introduced to her, Elaine Jones. And Elaine Jones was very famous in that she embroidered specimens of native plants and animals and made little dioramas out of them. She embroidered spiders and crickets and butterflies, miniature, and it's fascinating. And she said, I want to do pitcher plants. And so it was fortunate that I had with me photographs of all the species of pitcher plants. So I gave them to her, and she made this little diorama and shipped it to me in a box after I left. And so there's almost all 11 species of pitcher plants. There's flava and purpurea and maybe a Sarracenia in there somewhere, along with bog orchids, and there's an equisetum and other doodads that she embroidered by hand with her eyes, looking at them. It was fascinating. So I have this thing. It's about as big as a big basketball sitting in a display case at our botanical gardens. It's the most remarkable thing I ever saw. I don't know what's going to happen to it as the future goes on, but it's fun to think that someone would go to the trouble to do that. You see how evocative carnivorous plants are. They bring out, I think there's a Venus flytrap in there too. They bring out the artistic flair in people. So the issues I want to talk about today very casually with you, these are just hypotheses or just ideas in my mind. Given that Sarracenias must cross-pollinate to make seeds, this is true. Theoretically, there's no self-pollination in pitcher plants, but things can happen. And given that the pollinators are, as far as we know, bumblebees, no one's gone out and sat in the field for reasons I enumerated to observe for very long. These bumblebees that pollinate the flowers can also become prey later on. Then we should ask the question, how does each species of pitcher plant avoid eating its pollinators? And Andreas did a fabulous job yesterday. Fabulous. Couldn't have been a better introduction, because I don't have any of the technical slides he had showing how insects visiting various carnivorous plants get the nectar, affect pollination, how they may or may not be distracted by the trapping mechanisms, because the species do need to be a little bit worried about their pollinators being distracted by what appears to be nectar, what actually is nectar, in some parts of the plant. So how do pitcher plants avoid eating their pollinators? And why is this important, and what sort of background do we need? Well, it's important because if you're studying the natural history of these carnivorous plant genera, pollination is pretty important. So some people should, hopefully in the future, some of you all will do some field studies on pollination in Sarracenias, if there's any left in the future. So some of the ways that the pitcher plants avoid eating their pollinators. Here's an enumeration. Some of the insects won't fit in the traps. And so, for example, I love these pictures. Part of this talk is an excuse just to show you pretty pictures. I couldn't believe this when Robert Coe, several years ago, showed me, this probably happens all the time out here in California, hummingbirds eating the nectar of a pitcher plant. So I'd never seen that before. And they certainly aren't going to crawl down in that tube. Butterflies can't fit in a Sarracenia minor. Dragonflies don't even eat nectar. Here's a monarch butterfly. It certainly can't. It's feeding, it's getting some food, but it's not going to become a victim under the usual circumstances. So of course, these are also not pollinators, but they're not going to fall in the tubes. You've got praying mantis. I love this. This is my signature picture of a little frog down inside of a Sarracenia tube. When Mark Catesby, the famous English naturalist, first came to South Carolina in 1712, stayed here for about 15 years, he painted the first pictures of native plants in the East accurately. He wrote this huge book on the plants of the Southeast. He wrote when he published this in 1745, he wrote, God made pitcher plants as a place for insects and frogs to get in out of the sun. And that's all, that was the dogma for the next 100 years. And so that little guy there must have read that book. And of course he knows what's happening. He knows this insect's coming there, and so he knows he's got a free meal at McDonald's. And I don't think the pitcher plant cares, because the little frog is leaving some organic matter behind. So the pitcher plant is getting his dues by allowing the frog to be. But you know about the spiders, you know about the wasp. They hang around. They may get some nectar. They probably are catching. The pitcher plant is a microcosm of activity. It's like It's like like a little town. And you've got people rushing off to work. You've got people collecting what they're collecting. You've got people standing outside the bank, bugging people who come out. You've got back alley. You've got people jumping on you in the back alley. You've got people setting traps. Everybody is making a living from the pitcher plant, who's created a focal point for activity of all kinds of eat and be eaten, and be a victim or be a what's the opposite of a victim? But you know what I mean. A whole lot going on there. And then of course Sarracenias do attract, catch, digest, and absorb. Those are the 4 hallmarks of a carnivorous plant. They have to do all 4 of those. They eat mostly insects and small arthropods. They occasionally eat a little frog or a lizard, I suppose. But it's the mouth of the pitcher and the hood and the opening there. Let me go back. The opening there that secretes this nectar when the pitchers are fresh and absolutely attract insects of all kinds. And some are braver than others. Some have the ability to hang on and get out, and others don't. And then victims, of course, fall in and are digested and absorbed. This is one of my favorite. This is a fly contemplating suicide. This was the first famous picture I ever took back about 1971 or 72. So I've been using this picture for all these decades. And it's true. And these plants absolutely attract prey of all kinds. And the pitchers work. They fill up with insects through the season. You've got seasonal changes in who comes there and how they're interacting with each other. The first ones are coming looking for nectar. They fall in and die, and they start smelling bad, so they attract the ones who are coming there looking for dead meat. And they attract the ones who come there to ambush the ones who are coming there for the dead meat. And so you've got a seasonal changes of what's being attracted to pitcher plants. These can be studied. I had an honors student in 1995, Andy Baldwin, who studied Sarracenia flava. He went out and collected 250 tubes, which would be in flavas are not long season. They sort of finish up in midsummer. So he went out in midsummer and collected tubes, and he meticulously cut them open, slit them open, carefully removed the insect contents, carefully identified them as best he could. He had had a couple of entomology courses, and he did lots of data. This is sort of a summary that shows the array of insect prey caught in these tubes. Coleoptera are beetles. There are a lot of those. Hymenoptera, those are flies and bees. There are a lot of those. Lepidoptera are moths and butterflies. There are a good many of those. Hemiptera are the true bugs, like assassin bugs. Diptera, those are flies. Homoptera, those are leafhoppers. Spiders, those are spiders. Orthopteras are grasshoppers and crickets. Praying mantises and the like. Collembola are the microscopic insects that probably live down in there and help eat some of the prey. And then Neuroptera are pretty rare. They're a tiny little slit right in there. Those would be like damselflies, mayflies, fish flies, and other things that come out at night. And who knows why they end up in here. So a whole array. And by looking at them over the season, he showed that different ones were coming there for different reasons and in different. And believe it or not, about 10 years ago, back in North Carolina, they used Sarracenias as biological monitors to measure the invasion of non-native ladybugs. They would put Sarracenia clumps around town, around Raleigh, North Carolina, and trap is like putting out a trap for gypsy moths or other invasive insects that the Forest Service might be monitoring. They use pitcher plants as these collecting traps to see, because ladybugs would just flock there. And that was fascinating to see that study. And so Charles Darwin didn't know that Sarracenias were carnivorous. That's hard to believe because we thought Charles Darwin knew everything. But in 1865, when he published his book on insectivorous plants, that Peter talked about the other day as kind of the beginning of carnivorology, he didn't know pitcher plants were carnivorous. And he didn't include them in his book. He didn't even mention them. He didn't even know about them. And in 1875, an ancestor of mine, Joseph Mellichamp, who lived in South Carolina, he studied Sarracenia minor. He was the first one to prove and publish the facts that pitcher plants were carnivorous, and that they eat ants, especially. I think they like the black and red ones with taco sauce on the side. More about that later. But so in the second edition of Darwin's book, published by his son, in 1893 or so, the second edition of Insectivorous Plants was a footnote. And the footnote now, talk about being a footnote to history. Here's a footnote. Dr. Mellichamp and Dr. Candy, there can scarcely be a doubt that Sarracenia and Darlingtonia may be added to the class, that is of insectivorous plants, though the fact can hardly be considered as yet fully proved. So you see how slowly science can move sometimes. So even though he mentioned it, he was skeptical of it. And so that's my little footnote to history there. And so you know how beautiful pitcher plant flowers are. They can be red or yellow. And you can maybe identify some of the species by their flowers. Flavas and Oreophilas have long, narrow petals, and the odor of cat urine. Rubras have very stereotyped flowers, all members of the rubra complex. They have these funny little cupped sepals and these little small, broad petals, and they usually smell sweet. Hence, sweet pitcher plant is their common name. But other pitcher plants don't seem to have any very strong odors. I certainly would be interested in receiving observations from other folks. These are Eleda flowers. And so when you start mixing them as hybrids, they come out in different shades of reds, yellows, pinks, oranges, and other colors. But these are the sort of stereotypes of Sarracenia flowers. And one day when I didn't have anything else to do, when was that? It was 9 a.m. on June the 4th of 19. I decided to pick apart a Sarracenia flower. So I encourage you all to do this. And they're the bracts, they're the sepals, they're the petals, the style, the ovary. I don't know how many stamens. No nectar. I didn't find any nectar. So I'm not sure Sarracenias have a whole lot of nectar to offer. But the picture Andreas showed of the unique upside-down flower of Sarracenia with that umbrella-like style, or technically umbraculate. The flowers hang upside down. The pollen falls into the umbrella like pennies from heaven. If you open your umbrella, you collect pennies from heaven, and the bees go in there and they wallow in that pollen to get the pollen. And that must be pretty good pollen, or at least abundant, because they like to do that. No one has really studied in the field of pollination biology. My best, I suppose, picture is this of a bee coming out of the flower. I don't have a picture of a bee going in. They move pretty fast. They're hard to photograph. You'd need a pretty good, someone should take a pretty good high-speed photograph of pollination. They should perhaps, as Andreas did, pick the petal away so you can see. There's some beautiful photos on the internet of the inside of fresh flowers and what they look like down inside. If you're going to do any cross-pollinations, you need to know that pitcher plants are protogynous. That is, the stigma matures first and has to be pollinated by an incoming bee, and then a day later the stamens shed pollen, so the plants really can't be self-pollinated because they aren't both sexes at the same time. So there's a picture, as we have seen, of the cutaway of the flower. There's the stigma. The bees have to come in over the stigma, and then they enter by lifting up the petal and going out without touching the stigma with its own pollen. Pretty ingenious for a plant to figure that out. But the pollinators, so I don't know how to say this, the pollinators may be sacred to the plant at the time of pollination, but they will gladly eat you when you come back later. So these bumblebees are just as susceptible to the nectar of the pitchers after pollination as anybody else. This big bumblebee kept going down into this leucophila, the downward pointing hairs keep you going down. The nectar is all in there, and there she falls in and can't get out. I helped her out twice. And then I said, I'm tired of helping you out of here, you're on your own. So I would put a straw down in the tube, and she would crawl out on that straw, and she'd come right back. So I said, this is sort of like a drunk guy in a bar. You chase them out and say, go home. Those of you who were out last night probably know about that. Go home. No, I just need one more drink. It's so wonderful. So there's no question that this nectar is highly desirable to the insects. I think that's part of the strategy of the pitcher plant to make the nectar they secrete very desirable. So if the pitchers are effectively trapping and catching all kinds of insects, especially pollinators, what keeps the pollinators from being caught at the time of pollination? How do they keep from being distracted? That was Andreas's theme, I think, yesterday. In other words, don't shoot the messenger. Your bee is your most important part of your life cycle, transferring pollen. That's sexual reproduction. It's vital. It's the only way you're going to make seeds. And so you don't want to disrupt that. Do pitchers want to keep their pollinators to use them again? Or do they want to eat them and prevent them from others being used by other pitcher plants as a form of interspecific or even intraspecific competition? We don't know that. So that's my hypothesis is that it's better not to eat your pollinators and thus to use them again, let them be on their own. Can't get any seed if you don't have a good array of pollinators. And therefore, pitcher plants have evolved mechanisms or strategies, if I can be anthropomorphic. They've evolved strategies to prevent eating their pollinators. So how does each species do it? I realize this is not a scientific survey. They might not mind eating their pollinators after the act of pollination. I mentioned that, and this has not been studied. So someone should look into that. So some ways to avoid pollination capture. As I mentioned earlier, the size of the tube might not fit the pollinator. I don't think a rubra could eat a bumblebee. The tube isn't big enough. I don't think that a miner, I don't know if bees are attracted to that covered up hood of miner, even though the tube is big enough. I don't know if a bumblebee would fit down in a wherry flower, tube, I just don't know. Then there could be the relative timing of appearance of the flowers and the traps. So the flowers could come earlier and the traps could come up later. Or the traps could come earlier and the flowers bloom later. You've got examples of those, so you could have a difference of timing. And then the relative height of the pitcher trap and the flowering stalk could be different enough to hopefully keep the pollinator from being distracted. Tall pitchers, tall flowers. Flava, Oreophila, Leucophila, Elata, some members of the rubra complex. The pitchers and the flowers are about the same height. So one could have a hypothesis that even though the tubes are up, they're not functional yet. Maybe they haven't secreted nectar yet. And sometimes you don't see them that well developed when they're flowering. So the flowers and the pitchers might not. And Flava and Elata, they bloom much earlier than their pitchers. This particular picture, there are no pitchers yet, but you have flowers. And so you get different degrees of timing of the pitchers appearing. And of course, no distraction here, less chance for catching pollinators. That'd be the ideal way, I suppose. There's Oreophylla. Oreophylla, and you may find it in bloom and with a pitcher open, but is that pitcher functional yet? Has it secreted nectar? I don't know. I haven't studied these. Leucophila, most wonderful and beautiful. The flowers may be blooming when the pitchers are coming up, but Oreophyllus, if you've studied those even in growing them in cultivation, they tend to attract moths. They don't as often attract other things, although they certainly can. So these pitchers mostly eat moths in the late summer when these light, areolate pitchers at night reflect moonlight, perhaps, and look like candles in a meadow to moths. I've been out. Another thing y'all should do before you die, go to Splinter Hill Bog in in east Alabama and sit out there one night. I don't know what might happen to you. And just...
I did that.
Exactly. You are the one who has. I can't remember whether I have your picture of the moth. Damon published, I think, a most unique photograph from Splinter Hill Bog of a moth, an actual moth, flying and visiting an actual pitcher plant. He got an actual photograph of it. And that's what happens when you sit out there at night.
The mosquitoes don't bite me. I'm too bitter.
Okay. That's very good. So I don't have any pictures of that, but that's something you should try to do the next time you're east in the southeast. The rubra complex, this is Jonesii, tall pitchers, tall flowers. I'm not sure the pitchers are quite functional yet at the time of pollination. The mouths are awfully small. Whether a bumblebee would even want to go in there or not, or could if she wanted to, don't know. It's possible. It might not be likely. Purpureas, there's no question that in some of these cases, as Andreas mentioned, the flowers are tall, not so much to keep the pollinators away from the pitchers, but so the flowers can be seen by the pollinators. Because these plants are often down below the grass if it hasn't burned. Even in the northern, this is a northern type, growing in a pitcher plant bog or a wet sandy beach, and the flowers stick up high, perhaps so that they can be seen better by pollinators. But it could be that they're trying to get away from being close to those pitchers. Don't know. Have to do some experiments. Same with Sarracenia, where the pitchers grow low down, even underwater, and the flowers are tall. Pitchers and flowers may be functional at the same time, but flowers are held well above. An exception to that is Rosia down the Gulf Coast. Little short pitchers bloom early in the spring. My feeling there is that these pitchers are not functional yet. They haven't wakened up. They haven't waked up from their winter rest, and often they're dried up from the winter because they're fairly delicate, fairly thin pitchers. So I would say there that they're normally not, pitchers are not functional when the flowers are blooming, and so being tall is not of any great value in the sense of separating the flowers from the pitchers. Sarracenia, they grow short, send up tall pitchers. They often eat things underwater anyway, or down on the ground, ants and other aquatic things that crawl in through those very difficult to get to holes in the pitchers. I don't know if a bumblebee would be able to go down and even find that if they wanted to. You cut them open, they're full of these gigantic downward pointing hairs. If you've never opened a Sarracenia pitcher, you should. They have the longest hairs, and boy, aquatic insects really get caught down inside there. Sarracenia huerii, pitchers open, maybe only flies go to those. I don't know when they're first open. It's easy for the pollinator to get in the tube there if it wanted to. Rubra, look how tall the flowers are. That's a distinctive trait. Its flowers are really tall compared to the size of the pitchers. Minor, their pitchers are short. They come along late, long after the pitchers have been up, and so the flowers are shorter than the pitchers, and these things eat ants anyway. Ants climb up the tube, and they look in there and see those lights, those glistening, gleaming. It's like the Crystal Cathedral in California. But there's no salvation in there. They think it's the light show at Disneyland, they just jump right in. And they fill up with ants. You have not, so another thing you need to do before you die, you have not lived until you have collected some Sarracenia minors down around the Okefenokee, those really tall ones, to take back home to grow. And you've taken them and laid them carefully in the back of your car, and they are still full of live ants. Those ants are not dead yet. And the next day you come out to your car, and there are ant trails all over the inside of the car from the 10 million ants that were in the pitcher tubes. So we need to do that sometime too. It took me a week of vacuuming out the ants. Talk about taking your work home with you. So here's a beautiful picture of the tall minors with the short flowers. The pollinators are probably not going to be easily distracted at that level. And so finally, my favorite one of these is Alabamensis. It's been an enigma. One of the traits of Alabamensis, Rubra alabamensis, is that it comes up early, has big pitchers, and it has tall flowers. And the early pitchers, when the flowers are blooming, fall over and become these S-shaped or sigmoid pitchers. So I think that it could be that those tall pitchers that come up and then lay over are a way of avoiding having the pitchers up in the same zone as the flowers when the flowers are functional. It's a way of moving your pitchers out of the way at the time of pollination. So that's my explanation for those sigmoid pitchers that are so distinctive on Sarracenia alabamensis. So here's another picture of that. Here are these early spring large pitchers. They're functional, laying over to the side. This is a cultivated plant, so it might not, in the wild, I don't think the flowers get that tall. They're up around the same level as the pitchers. So the sigmoid pitchers laying over to the side. So what to do about this hypothesis and these problems? Well, the usual things. Somebody needs to go study them in the wild and in the test garden, and do some artificial manipulations using pitchers and flowers and see how bees relate to that. So in closing, this slide and one other one, in this world of eat or be eaten, and the bizarre behavior of many plants to do the unexpected, the study of various adaptations provide interesting twists and turns. And, this is why we study them. It gives us challenges to figure out these puzzles of nature. If you haven't seen, now it doesn't have pitcher plants in it, but if you haven't seen the PBS video, What Do Plants Talk About, you should see that, because it is a modern view of the bizarre things that plants can do as a form of plant behavior that we never thought about. So my last idea to you all is about the future, and for you young people in the audience. This is a little history of the monographs, that is the taxonomic write-ups summarizing Sarracenia species and how many there were, what they were, and what descriptions there were down through history. In 1914, John McFarland in Bailey's Cyclopedia of Horticulture, that was the first enumeration in a semi-technical way of the pitcher plants known at the time. And then 1930, Edgar Wherry wrote illustrations in a publication called Illustrations of North American Pitcher Plants. He enumerated what was known, and for the first time drew maps of their distribution. And he recognized things like Oreophila for the first time, and Jonesii, although he didn't call it Jonesii at that time. In 1949, Richie Bell, professor at UNC Chapel Hill, wrote his PhD thesis was the genus Sarracenia. So he had an enumeration of all the species. In 1971, 1971, Sidney McDaniel wrote a paper on Sarracenia, which enumerated all the species and had dot maps for where they all grew. And then 2009, Mellichamp and Case, the late Fred Case, and I, mostly me, wrote up treatment of Sarraceniaceae for flora North America. So that's my opinion, and that's the latest formally written opinion. And of course, there are other opinions. So what I'm bringing to you all is it's been on average of every 20 years that somebody's done a monograph to capture what's the latest in Sarracenia. So the next one might be due about 2030, and who in this room is going to do that? One of you all need to grow up and become competent in Sarracenia, I don't mean it that way, become competent in Sarracenia taxonomy so that you can do the next monograph in 2030 and deal with some of these problems. Like, what is going to be the Sarracenia rubric? How does the Sarracenia rubric complex really shake out as we learn more about molecular biology, perhaps? Fernando is going to talk about that later. The Sarracenia purpurea complex, which seems like a simple thing, but we don't know what Montana is. It needs to be better described and identified and located and see how it fits in. I think it's just a dwarf local form of purpurea, but who knows? And we'll be better able to work out perhaps the evolutionary history and relationships. Since they all hybridize, it's going to be difficult because of the fact that none of them are pure. Maybe they're all hybrids anyway. I don't know. So that's a challenge for the future for you all to be thinking about as you study these plants. And so that's the end. Since I started late and talked too long, there's not much time for questions, but does anybody, and I'll be glad to talk with anyone. Does anybody have a question? Yes, sir.
Just in addition to Sarracenia, it does have nectar.
Okay.
This nectar in the flower, there's a nice presentation that's
Okay.
all about the nectar.
Okay. Yeah, you mentioned that, and so I appreciate that, and I hadn't discerned a whole lot of that. Thank you. Yes, sir. Okay. Are they good? Okay. Let me just say, Mike was saying there are 2 publications on pollination, and I accept that. I'm an old guy and retired, and so don't keep up with the literature. And so if you take my comments for what they're worth as ideas and hypotheses and things to mull over, rather than definitive pronouncements.